Scientists just found human DNA in a place nobody had ever recovered it before, inside the surface of a cave wall. Not in a skeleton, not in a tooth, not even in the dirt below. The genetic trace was trapped against rock, beside prehistoric pigment, after the person who left it had disappeared for thousands of years. It sounds like the perfect way to identify a cave artist. But the discovery comes with a twist. The wall remembers a human. Science still has to prove what that human was doing there. The breakthrough was announced in June 2026. An international team had sampled caves across Spain and Portugal between 2022 and 2025. They visited 11 caves and studied 24 panels containing simple red marks, hand stencils, and famous animal paintings. Their question was almost impossible to resist. If ancient artists touched the wall, rubbed pigment with their fingers, or blew paint from their mouths, could a few cells have survived with the art? Until now, ancient human DNA usually came from bones, teeth, artifacts, or cave sediment. Each source has limits. Bones are rare. A tooth may belong to someone who never made the objects around it. Dirt can mix traces from many people and animals. A painted wall is different. It is the exact surface where a human once worked. If that surface kept their DNA, the artist might have left something more personal than a picture. The researchers entered the caves wearing protective clothing. Modern skin, hair, breath, or dust could overwhelm the ancient signal, so every movement mattered. At selected panels, they removed tiny pieces of pigment or the thin calcite crust that had grown over it. They also sampled nearby bare wall as a control. In one case, they took less than 1 milligram of pigment from inside a prehistoric bone tube once used to blow paint. Then the samples traveled to clean laboratories. Researchers dissolved the mineral material, built DNA libraries, and searched for the short damaged fragments expected from ancient molecules. Most samples gave them nothing useful. The bone paint blower from Altamira did not preserve an authentic ancient human signal. Neither did most of the painted surfaces. Out of 24 art panels, only one produced convincing ancient human DNA. That one success came from panel 11 in Escoural Cave, Portugal. A small pigmented calcite crust contained fragments of both mitochondrial and nuclear human DNA. The molecules showed the chemical damage that accumulates with age. They were not simply fresh DNA dropped by a researcher. For the first time, a piece of prehistoric rock art had yielded an authentic ancient human genetic trace. The strangest clue was what the sample did not contain. It held human DNA, but no detectable ancient DNA from other mammals. Cave dirt usually carries a busy mixture from animals as well as people. A clean human-only signal made ordinary sediment less likely. The most plausible explanation was direct contact. Skin, sweat, saliva, or another bodily fluid touching the wall or the material that became sealed against it. Picture the possible moment. Deep inside Escoural, a person prepares red pigment under a small flame. One hand steadies against cold limestone. Fingers rub the wet color, or a breath sends droplets toward the rock. Microscopic cells land with the pigment. Mineral crust slowly closes over them. The flame goes out, the person leaves, and the wall begins guarding a secret no one knows is there. That scene is possible. It is not proven. The DNA might belong to the artist, someone who prepared the pigment, or a later visitor who touched the same place. The pigment and the genetic trace cannot yet be dated closely enough to show they arrived together. They may have been deposited years apart, or even thousands of years apart. This is why the real discovery is more careful and more interesting than a simple headline about finding the artist. The control samples produced another surprise. Ancient human DNA also appeared on unpainted walls at Escoural and at Escoural Cave in northern Spain. Some of those traces were mixed with DNA from deer, cattle relatives, cats, rabbits, and other animals. That mixture points toward a different route. A person may have touched the cave floor and then braced a dirty hand against the wall. Water may also have carried old material through the rock. Together, 5 wall samples contained authentic ancient human mitochondrial DNA. 3 appear to contain DNA mainly from women. 1 is consistent with a man. The painted Escoural sample did not contain enough information to determine sex. Even these small clues change the feeling of the caves. The walls are no longer only surfaces covered with images.
They are places where individual bodies moved, leaned, touched, and breathed. 2 samples from Coveron contained enough nuclear DNA for a deeper comparison. Their genetic pattern aligned with Western hunter-gatherers, populations known in Europe from roughly 16,700 to 5,200 years ago. The result does not identify one named person. It does show that a bare stone wall can preserve enough scattered molecules to connect a human presence with an ancient population. How old are the traces? The answer is deliberately broad. DNA damage suggests most are at least about 2,000 years old and could be much older. Escoural gives a stronger clue because the cave became inaccessible around 4,000 to 5,000 years ago and stayed sealed until quarrying reopened it in 1963. Any human trace sealed inside must predate that closure. The cave also preserves Neolithic, Upper Paleolithic, and Middle Paleolithic activity, so several very different ages remain possible. That uncertainty protects us from inventing a face. We do not know the Escoural artist's name, age, voice, or appearance. We cannot say whether a woman or a man painted panel 11. We cannot claim that the Western hunter-gatherer DNA belongs to an artist at all. The honest picture is a series of possible human moments, connected to a wall by real molecules, but not yet connected to one exact act. And yet the method could become extraordinary. Many caves contain art, but no human skeletons. Others preserve footprints, soot, broken tools, or marks deep beyond daylight, while the identities of the visitors remain invisible. Sampling a wall could reveal which populations entered, whether men or women used different chambers, and whether the same people returned to particular places. A cave without bones might still hold a biological record of everyone who touched it. Future samples may go even further. Researchers want to test hand stencils, figurative paintings, and caves with stronger preservation. Some sites contain marks that may have been made by Neanderthals. If ancient DNA can be recovered directly beside those marks, one of archaeology's hardest arguments could move from the shape of an image to the biology of the person who stood before it. But success will be rare. In this study, 23 of the 24 art panels did not yield convincing ancient human DNA. A wall may be scrubbed by water, coated by minerals, handled by later visitors, or contaminated by the sediment below. The signal can survive in one tiny patch and vanish a few centimeters away. Finding it requires careful sampling, repeated tests, and the willingness to accept a negative result. That rarity is what makes panel 11 so powerful. Somewhere on a pigmented patch of limestone, a few damaged fragments crossed thousands of years. They survived darkness, mineral growth, cave closure, quarrying, excavation, and the touch of modern science. They cannot yet tell us who painted the wall. They can tell us that a human body was there. Imagine the cave after the researchers leave. The lights move back toward the entrance. Painted marks fade into blackness. Nothing on the wall looks alive. But inside one thin crust is a broken genetic whisper from a person who once touched, breathed, or worked beside that stone. The picture may have lost its meaning. The person's name may be gone. The wall kept one final secret. Somebody was here.